everyone, welcome to my channel, and today I come with another video, that is the chromosomal mutation. Generally two kinds of mutations are there. One is the point mutation and another is the chromosomal mutation. Generally the point mutation, that is the mutation occur in the base pair, DNA base pair, and the chromosomal mutation, this is the part of one chromosome that is either deleted or duplicated or inverted, anyhow. So this is the chromosomal mutation that occurs in the part of the chromosome, not the base pair. Clear? And the point mutation that occurs in the DNA base pair. Clear? So first today we learn what is the chromosomal mutation. Chromosomal mutation is the structure or the chromosomal numbers, the variation in the chromosome structure or the chromosome numbers, and that is found in the wild type chromosome. So actually when the number or the structure of the chromosome, normal chromosome or the wild type chromosome, that anyhow changes, then the chromosomal mutation occurs. Clear? So there are many types of, generally 4 types of mutations found in the chromosomes, and the first one is the deletion. What is the deletion? Deletion is the part of the chromosome that is missing. Here, part of the chromosome that is missing. Here is a chromosome, you just imagine, and there are the parts. Okay, A, B, C, D, E, F, G, H. Okay, and the deletion process is happening here. And what is that? The D part of the chromosome that is totally removed or deleted. Okay, you can see this is the chromosome, and after the process of the deletion, this is formed. Clear? And you can see here the D part is not found. Okay, A, B, C, D, E, the D part is missing. So this is the deletion. Okay, and this is possible, or the part of the chromosome that is deleted by some agents. That is radiation, heat, etc. Okay, these are the agents by which any part of the chromosome must be deleted. And if the deletion involves the centromere, that means you know that the centromere is actually this is the chromatid. Okay, sister chromatid. The two part of the chromosome join constriction, through a constriction, and there is present centromere. Okay, and if the deletion is involved with centromere, that means the centromere part is deleted with the chromosomal part, then what happens? The chromosome is called the acentric chromosome. Clear? This is called the acentric chromosome, and this acentric type of chromosome generally is lost during the meiosis process. Clear? And so lethal consequences sometimes is possible. Okay, lethal consequences may be created sometimes. And one disease of this deletion of chromosomal part, this is the cry-to-chat. Okay, cry-to-chat is one kind of disorder, human disorder obviously. This is a syndrome or disease. This is found in the if the chromosomal part is deleted, somehow this disorder is occurred in human. Clear? So this is the process deletion. Another process is the duplication. Okay, we learn after that deletion. I think you can understand what is deletion. Deletion is the part of one chromosome that is deleted or removed by any agents, due to presence of any agents like the heat, radiation, etc. So in that situation, if any part of the chromosome is deleted, then the number or the structure, both are changes in the chromosome or wild type chromosome, and that created many disorders. One example is the cry-to-chat. Next one is the duplication. This is duplication. Duplication is the doubling of the chromosomal segments, and it occurs at the different location in the genome. So here the parts or the segments of the chromosome that is doubled. Okay, that is doubled. So you can see here one chromosome and the segments are there, A, B, C, D, E, F, G, H, and you see the after duplication the segments are increased in number. Okay, here the BC segments that are duplicated. Okay, BC segment that are duplicated, and this type of duplication is called the tandem. Okay, tandem. Next one, if this type of duplication is seen, if this type of duplication is seen means here you see the segment is BC, BC, that BC is same, but after that the reverse of the BC is found. Okay, reverse of these two segments are found. This is the reverse tandem, and the last one is the terminal tandem. And here the ABCD, AB changes. AB changes. You see that AB and again AB. This is called the terminal tandem. Okay. So this is the duplication process. Duplication process is the segments of the chromosomes that are duplicated. So there are the three types of duplication is possible. One is the tandem, that means just BC and BC. Tandem and terminal tandem is same in such a way, but different that there is one difference is that the A is the terminal and this is doubled and again found A. Okay, so this is the terminal. Okay, so and this duplication example is generally alpha and the beta globin. The sequence of the alpha globin and beta globin is always similar in case of hemoglobin structure, always similar, but it is thought that the all type of the beta and alpha globin is not similar in all the individuals. Okay, so this is possible. This divergence or the dissimilarity, sometimes it is possible the duplication due to the duplication process of the chromosome. Clear? So this is the duplication. Next one is the inversion. Okay, inversion. Okay, this is inversion. What happened in inversion? Here the segments of the chromosome is excised and then reintegrated at an orientation 180 degrees. You see here the segmentation generally it 180 degree. It is first excised and then reintegrated at orientation of 180 degree. That means it reverses in such a way that the 180 degree inversion is seen here. And what happened? You see the structure. A, B, C, D, the segments is proper segment. This is the wild type and the normal chromosome. And here after 180 degree integration, what happened? You see that B, C, D, here D, C, B. Okay, and after that E, F, G, H is proper. Okay, then what is the inversion? Inversion is actually is mentioned obviously the 180 degree inversion is possible. That means the first of all it excised and then reintegrated at 180 degree and then joined to each other. Okay, so here two kinds of paracentric and pericentric inversion. In case of paracentric, the centromere, you see that centromere it is a place where the centromere is present and to join the chromatid, sister chromatid. So you see here the inversion is possible in here. B, C, D segments, not here. So centromere is not included here. Okay, so centromere is not included. That means it is the paracentric inversion. And in case of pericentric inversion, here the centromere is included. You see here the centromere is present here and the inversion is occurring D, E, F, and you see here F, E, D. The position of the centromere changes. Okay, the position here is the position, this one, and the position changes here. Okay. Okay, clear? So this is the inversion. And what disease or disorder is found here? That is the acute leukemia. Okay, acute leukemia. So inversion is first excised and the inversion of 180-degree orientation and then integrated with the chromosome part or the segments. Here, this is the inversion. And the last one is the translocation. This is the last one. Okay, this is the translocation. Translocation chromosomal mutation. What is this translocation chromosomal mutation? There is a change in position of chromosome segments. Okay, position of chromosome segments within one chromosome or between the two chromosomes. Clear? The segments are changes. Its position. Okay, this is the translocation process. So here, in case of translocation, we see one is the non-reciprocal translocation, another is the reciprocal translocation. Clear? In case of non-reciprocal translocation, there is the exchange of segments is seen only in one chromosome, within the chromosome. You can see here, these are the segments and the BC part that is exchanged with this, the lower part. Okay, you see here A, B, C, D, E, F, G, H, and here this changes and its position. BC changes its position. Here in the lower, it transfers to the lower chromatic part. Okay, and here A, D, E only present, and here the BC is present. Clear? So this is the non-reciprocal intra-change. Okay, intra-chromosomal mutation. That means within the exchange of segments within one chromosome or the single chromosome. This is the intra. And non-reciprocal inter means here the exchange of segments is possible between the two chromosomes. But here only one, that is the segments of one chromosome that transfer to the other chromosome. But here the segments of the other chromosome is not transferred to the previous one. Clear? That's why it is a non-reciprocal. That means this is the not the changes is bidirectional. That means the changes is possible is in one direction, or only one chromosome exchange their segments. This is clearly said that one chromosome only exchange their segments here with the other chromosome, but the other chromosome here remain try to remain in the same. And you see here ABC, the BC part is changed or transferred to the MNPQ, that is BC here included, and but the segments of the second chromosome, they remain in the same position. Okay, same position means they remain actually in the same chromosome. Okay, they remain in the same chromosome. There is not the parts of the segment that transfer to the previous one. This is the non-reciprocal translocation, but it is called the inter. That means the exchange is possible between the two chromosomes. So that's why it is called the inter. In case of reciprocal translocation, here the exchange of the segment is possible between the two chromosomes. That means the two chromosome means one chromosome exchange their segments to the second one. The first one exchanges their segments to the second one, and the second one also transfer their segment to the first one. Clear? You see here AB that transfer to the second one, MN position. Okay, and NO, these are the segments of the second chromosome that transfer its segments to the previous one. You see here. So that means the translocation means the position of the segments that changes its places. So reciprocal and non-reciprocal means here reciprocal means the one chromosome only exchanges their segments, and the second one, or the in case of it happens within same one. So the chromatid part, the two chromatid attach with the centromere to form the chromosome. So one chromatid remains same, or the segments of one chromatid remain same, but the other segments of other chromatid that places or that exchange their places. Clear? So this is the intra, or this is the reciprocal way, non-reciprocal way. And in case of reciprocal way, there both the chromosome exchange their parts. Clear? The non-reciprocal means one chromosome exchanges their part or one chromatid exchanges their part, but in case of reciprocal, both the chromosomes, they are involved in exchanging their segments. So these are the okay, and the last one, this is one kind of disease is found due to this translocation process, and this changes of the places of the segments that occurring, the changes of the proto-oncogene to the oncogene. Okay, that is the non-cancerous cell that transfer to the cancerous cell due to this chromosomal changes. And one example is the Philadelphia chromosome, that is important or it is discovered. And why it is known as the Philadelphia? Because it is discovered in Philadelphia, and this is the example of this is discovered as the translocation chromosomal mutation. Okay, so these are the changes. So today we learned about the chromosomal mutation and their disorders, that due to the chromosomal mutation what disorders occurs in humans. Okay, so we learned here. I think you can clearly understand about this topic, and if you want to get more knowledges or help to me to provide you knowledges, so then you have to subscribe my channel, otherwise this channel will be not continued in YouTube. Okay, thank you.